Hey guys, so we talked about active versus passive transport in our last video. And so we really got all the information about active taken care of. And remember, it's these things. It requires energy. It's from low to high concentration. It is super great for pumps, endocytosis, exocytosis, all of those really awesome things. Okay, and big thing there, it requires energy. Okay, so we also talked about the passive types. So we talked about diffusion and all the different types of diffusion. So we got facilitated diffusion, which requires a protein. We have dialysis diffusion, which is just diffusion of particles. And then we mentioned that osmosis was diffusion of water. Okay, so I can't stress to you how important osmosis is. It is definitely going to be on the AP exam. It's going to be in a lot of different forms in the AP exam. This is a great kind of topic for us to make sure that we have a good, thorough understanding of. So for osmosis, the big thing is that yes, it is the diffusion of water, and water diffuses across a membrane, and it is passive, so it's going from a high concentration to a low concentration. Okay, so high concentration of water to low concentration of water. Okay, but water's kind of funky because in order to make that concentration a thing, there are concentrations of solute also in the solutions, okay, that have water in it. So what water actually is doing, it's moving towards a higher concentration of solute, okay? Still a lower concentration of water, so still passive, moving from high to low, but that water is also moving towards a higher concentration of solute, okay? So what that means is water is always just trying to dilute solutes, okay? Whether that be sugar, whether that be salts, any of those things, any of that stuff that can be a solution is a solute. Okay, so here's a great little visual. This is called a U-tube right here. A couple things that we notice. First off is we have 1, 2, 3, 4, 5 little dots, little pieces of solute on this side of the U-tube, a semi-permeable membrane separating this side from this side, which has a higher concentration of sugar. Okay, so the first thing I notice is that these green sugar particles are too big to fit through the pores of this membrane, this selectively permeable membrane. Okay, and so here's what's going on. Over here, believe it or not, we have a low concentration of solute, even though it looks like our water levels are the same. Okay, because we have a lower concentration of solute, that means there's actually more water proportionally to this side than there is this side. Okay, so over here, we're seeing a high concentration of these sugar particles, which means the proportionally there is less water to sugar ratio. So we've got water wanting to actually move across that membrane so that it can dilute this side of the U-tube. Okay, so over time, we can see that there's passive movement. The water goes to this other side, but this now is actually in equilibrium, okay? Believe it or not, even though it looks like it's higher over here, the concentrations are the same, okay? So our ratio of solute to water is the same, which is what it means by it wants to dilute. So let's talk a little bit, okay? We're going to harp on our water balance of cells without cell walls. Okay, so these are animal cells. We're definitely talking about animal cells here. And so when we talk about the balance of water, we like to talk about this word called tonicity. Okay, and so that's just basically saying it's water's ability to lose or gain water. Okay, so the three types of tonicity we have are isotonic, okay, hypertonic, and hypotonic. Okay, so I'm actually going to flip and show you a picture because I think it helps us understand just a little bit better than me just reading off this slide. Okay, so here is our animal cell. All right, we are going to move me over here. Okay, our animal cell is sitting in different solutions: hypotonic solution, isotonic solution, hypertonic solution. Okay, so in our animal cell, when we talk about hypotonic solution, okay, we have a solution where the concentration is low on the outside, boom, 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 and high on the inside of the cell. In an isotonic solution, the concentration of solute is equal on both sides of the cell. And then in a hypertonic solution, we have a high concentration of solute and a low concentration of solute, okay? So here's what that means. What an animal cell wants to be is normal. It wants to be isotonic. It wants to be in the same environment that is the solution of the cell. So when we're isotonic, we've got water coming in and out at the same rate, and our cell is able to stay the correct shape, okay? If we enter an environment that does not have as much salt as our cell, okay, water wants to dilute that cell, so it's going to go into the cell, and we're going to make this cell get really, really big, and then eventually it's going to pop, okay? So bad news bears there. We got a popping cell. We call this lysed, okay? We talked about hydrolysis. We talked about how it breaks things down. So that word lysed just means to break, okay? We also have hypertonic, okay? So if we take this normal cell, we move it to an environment where there is more salt on the outside. Hyper means more, okay? Hypo means less, okay? So more salt in the solution on the outside in the environment, okay? Water wants to dilute the solution, so it's going to move out of the cell. Boom, boom, boom. It's going to cause our cell to shrivel, okay? So for an animal cell, this is actually considered a normal cell, okay? So isotonic, moving in and out, equaling equilibrium, okay? Dynamic equilibrium to be specific, okay? In a plant cell, okay, so now we're going to kind of talk about osmoregulation, okay? In a plant cell, things are a little bit different, okay? So, in a osmoregulation, I'm not ready for plants, I got distracted, okay? Hypertonic and hypotonic environments create osmotic problems, okay? So if we take an organism that does not have a cell wall, like a paramecium, and we put it in a environment that is hypotonic, okay? So I want you to look at the way that this is phrased so that you don't get confused on a test, okay? If a paramecium is hypertonic, okay, that means the paramecium has the salt, okay? Which means the reverse is that the environment is now hypotonic, okay? So that's kind of confusing. They like to make sure you know, if you know hypertonic means salty and hypotonic means not salty, we can kind of decipher the way they phrase it a little bit better. Okay, so which is hypertonic to the environment, which would be hypotonic. We need a contractile vacuole to act as a pump and regulate that water balance. Okay, so we have this process called osmoregulation. These are adaptations that help us control the water balance. Okay, so here's our picture. We have a paramecium. He's nice and full. He has a hypotonic environment on the outside, so less solute. Moving into the cell, there's more solute, so we want to dilute. Water moves into the cell. Okay, it fills up this contractile vacuole. Okay, it fills up our paramecium, but then once he gets too full, instead of popping, he will contract, expel all of that water, and then boom shakalaka, he's like slimmed down again. You can see that the cell is not as full. You can see that the vacuole is empty, and that's how he controls his water regulation. Okay, if we are a plant cell, so we have a cell wall, we're a little bit different. Okay, here's the way we osmoregulate. First off, the cell wall helps us maintain a water balance, okay? Plants love to be this thing called turgid, okay? Turgid means that they are filled with water, okay? So I'm going to go back to this picture, okay? In a plant cell, normal or isotonic, I should say, isotonic is not good, okay? Water in and out at the same rate. This cell is not as full as it could be because this cell wall will not let this cell pop. Okay, so if we're in dynamic equilibrium, we're not as good as we could be. We're going to be something called flaccid. Okay, if the concentration of the cell is higher to the water on the outside, and by concentration I'm talking about the solute concentration, okay, then water is going to move in, and that cell wall is going to prevent the cell membrane from lysing. Okay, so it's not going to pop like it did in the animal cell. So turgid is the word, and this is actually normal for a plant cell. Okay, if the other side of that, if the outside environment is too salty, okay, it has high concentration of solute, more so than the plant itself, the water is going to move out of the cell and it's going to do this thing called plasmolyze. Please make sure you know this word, okay? What it means is that the cell membrane has actually pulled away from the cell wall. So this cell is not going to be able to transport nutrients the right way, get things into the cell membrane, be able to access those other organelles because of how empty it is, okay? Because this cell membrane is not directly touching the cell wall, so it has no way to kind of transport those materials, okay? So we can go back. Here is the plasmolysis. So not pictured here, but that was the third thing in a hypertonic environment, hypertonic salty environment outside. Okay, water moves towards the hypertonic always, so it's moving out of the cell and it's making our cell plasmolyze or pull apart from the cell wall. All right, so we have water potential coming next. I will make a separate video for that because it's going to be a long one, okay? But I hope that helped with osmosis. You guys are awesome. Have a great rest of your day.